Yeah.
Fantastic. So Jimena's going to be identifying Alzheimer's disease relevant neuroinflammation models with single cell iPSC-derived microglia. So over to you. There'll be a slight noise when we hit 12 minutes, but great.
Okay, thank you. Hi, everyone. Yes, as Caleb mentioned, I want to understand how microglia respond to different stimuli, and particular iPSC-derived microglia, and how these responses are relevant for Alzheimer's disease. Why microglia? Well, you already know the genes that have been associated to Alzheimer's disease through the different GWAS studies are enriched in genes that are preferentially expressed in microglia, as shown here by this group. We aim to model human microglia following a protocol from differentiation of iPS developed by Sally Cowley's lab, in which this protocol aims to recapitulate the developmental origin of the microglia from the yolk sac. And what we did is measure the transcriptional responses of microglia to different stimuli, and before and after 24 hours of exposure of these different stimuli, including ATP gamma S, which is a very slow hydrolyzable form of ATP, to the combination of LPS and interferon gamma, which is the bacterial neurotoxin and the cytokine, and to the lipid immune modulator, which is prostaglandin E2. So we did this using SiteSec, and we end up after QC with about 20,000 cells, which are represented by dots in this TSNE plot and colored by the expression levels of two markers. One is the microglial marker C1QB, and the other one is a fibroblast marker called 1A1. And more than 97% of the cells that we identify show the consistent expression of both these and other microglial markers, with less than 3% of our cells showing the characteristic fibroblast markers. So we also observe a large segregation by the different experimental groups with controls and those cells treated with prostaglandin E2 at the top. And those cells that were treated with ATP gamma S were clustering towards the right, either alone or in combination with the other two stimuli. And those cells that were treated with only LPS and interferon gamma were clustering to the left here. We also observe a segregation by the line or by the donor from which the iPS-derived microglia were derived from. So we observe a fairly consistent response across our different biological replicates. Once we focus on only in the microglia population and we run principal component analysis, what we observe is that along the first principal component analysis, cells that were treated with LPS and interferon gamma at both 24 and 48 hours segregate along this axis. And along the second component, we observe cells that were treated with ATP gamma S segregating along that component. We further try to identify the genes that are differentially expressed in each of these in response to each of these different stimuli. And what we find is that the largest number of the genes differentially expressed detected was in response to LPS and interferon gamma at 24 hours. This was closely followed by the number of genes that are differentially expressed in response to ATP gamma S at 24 hours. When we focus on the protein-protein interaction networks of the genes that had the strongest fold changes in either of these three conditions at 24 hours, which are exemplified here in color in blue, those that are downregulated and those that are upregulated in red. What you can see is the direction of the change and the strength of the change. We observe a quite similar response between ATP gamma S and LPS and interferon gamma, and a quite milder response to prostaglandin 2 by comparison. We then ask whether these gene expression changes were relevant for AD. So we first had a look at genes that have been already shown to be differentially expressed in microglia from AD patients. This comes from two postmortem studies from the prefrontal cortex and from the entorhinal cortex that Gabriel already pointed out to these results. And what we did is basically look at the overlap between the differential expressed in microglia in AD and those that are differentially expressed in our iPS stimulated microglia. And what we found is that we found a higher than expected overlap between this set of AD microglia differentially expressed genes and those that are differentially expressed in our iPS microglia across all the different comparisons. And what you can see in the heat map to the right is the gene expression patterns of these overlapping genes, which tend to be changing in the same direction within the iPS-derived microglia, with a few exceptions, which are the CCL3, which is upregulated in most of the conditions, except those treated with PGE2, and SEP1, which is upregulated only in the conditions treated with LPS and interferon gamma, and is actually downregulated with PGE2. Conversely, you have ApoC1, which is upregulated with PGE2 and mostly downregulated in the other conditions. We then next ask, well, given that we don't have like a clear answer for here, which of the stimuli is more relevant, we turn back to mice where a previous member of our lab, Devika, performed a meta-analysis of the microglia from different mouse models of AD. What you can see after accounting for batch effects, she identified the first principal component that segregated wild types from most of the AD models of AD models in mouse, and particularly more severe models such as the 5X8AD segregated further along this axis. What we did then is trying to relate this model to more generally AD GWAS hits, given that these models are characterized mostly early onset models of the AD. What we did is look at the genes that are either upregulated or downregulated along this disease axis and try to see whether we could find an overlap with AD GWAS hits. And actually, we find a significant overlap between those genes that are downregulated along this disease axis and AD GWAS hits, including some of these are classics being 1 and ADAM10, for example. Not only that, we also try to relate these genes that are changing along this disease axis with AD GWAS hits through protein-protein interactions. And we actually find more protein-protein interactions between these genes and both the genes that are upregulated and downregulated with the disease axis. We then ask whether we could associate the microglia response that have been already characterized in microglia subtypes to our disease axis. These are microglia subtypes in the mouse model of APP-NL-GF model, which has an increase of A-beta accumulation with age. And what we did is aggregated the profiles from the different microglia subtypes and projected into our disease axis. And what we found is a really nice segregation of the different subtypes with homeostatic microglia towards the left of this axis, while the activated response microglia that is responding to the I-beta plaques towards the right of this axis, and particularly this transitioning state somewhere in between these two. We can also aggregate the data by the genotype, in this case, the wild type and the disease model. And we also observe a segregation along the disease axis when we projected here in principal component 1. But this difference is not as strong as that what we obtained when projecting the microglia subtypes. We further repeat the analysis, but now in the APP-PS1 model, for which there is also data for a TREM2 knockout. And what we can see here is that, again, when we project the different microglia subtypes, a difference between the homeostatic microglia, here shown in blue, and that activated response microglia. When we look at the genotype, we also observe this segregation of the APP-PS1 model going towards the right of this disease axis, while the knockout of the ApoE knockout are just slightly towards the left. So what we did the next is trying to see if there is any stimuli that we could apply to microglia to move them along this disease axis. And what we did is do this retrospectively, looking at the gene expression data of Cho et al., which they characterize in mouse microglia a large array of the response of microglia to a large array of different compounds. And we projected this data into our disease axis. And what we can see here in this orange in the middle is the response of microglia to LPS that are shifting along this axis. What we have done next is use our own human IPS microglia that for which we have the microglia responses and projected into the disease axis. And what we have found is that only in those stimuli that are containing LPS and interferon gamma, we observe a shift along this disease axis. So to sum up a bit, we have identified this disease axis that segregates the wild types from the ADA mouse models. And this corresponds well with known microglia subtypes, such as hemostatic and versus activated response microglia. And this allows us to identify LPS and some stimuli that can shift the microglia transcriptional profile along this disease axis. I just want to thank the organizers for the opportunity to present their work, our collaborators, Caleb, and everyone in the lab. Thank you.
Brilliant. Thanks, Jimena. Everyone's super on time today. I have an easy life. I don't have, that's great. So everyone, please put your questions into the Q&A or the panelists into the chat, and I'll try and pick them up from there. So obviously this is a workshop about the single cell and the solutions that single cell can bring. Now, in your experiment, you started off using, basically, you identified this subpopulation of fibroblasts, but it wasn't a big population, and everything else seemed to be reasonably sort of microglia-like. Obviously, these are iPSC models. Do you think that if you'd kept that fibroblast in it, i.e., if you'd done bulk instead of single cell, you kept that fibroblast population that you didn't like, you threw out later, but ultimately if you kept it in, do you think it would have disrupted your experimental findings significantly? I.e., do we need single cell to help us resolve iPSC model microglia heterogeneity?
If they don't, if they were not to respond to the different stimuli, then it wouldn't matter. But if they do have a response that is different from that of iPS microglia, then I think it is important to remove them. So iPS, well, the single cell will allow us to keep only the iPS-derived microglia, and I think that is particularly important because there are like some of microglial markers, such as TGF-beta, that was actually expressed in this fibroblast population. So this is also something that we can segregate by doing single cell sequencing.
So do you think even though it was a small population, you still needed the single cell approach to take apart the heterogeneity? Oops.
Yeah.
Brilliant. Okay. And then one final question. Oh, here we go. So this is a question from Veronique Miron. Interesting work and great talk, Jimena. Given your data on the LPS pathway activation and microglia signatures, can you link this with infection and gram-negative bacteria? Is there data on this that you could link in with your data?
I mean, yes, what we have done is try to, well, what is really just studies with LPS again in mice. But not necessarily with the bacteria per se, but I think it's quite a good proxy of what you will expect with the bacterial response. But yes.
Because there's this open question, isn't there? I mean, and you know, about the role of infection in the brain, its association with dementia, and there seems to be more suggestive results saying it might play a part. And you know, your alignment of obviously LPS lipopolysaccharide is a bacterial antigen. So so do you think it adds any evidence to say, well, perhaps the response that we're seeing from brain infections could have something to do with provoking Alzheimer's-like pathology?
It could be. What I could say is that at least we also compare what I didn't show in vivo response to LPS, which is also kind of a systemic exposure in mice. And we see a great commonality in the pathways that are coming up and down between the response in the iPS in vitro. So yeah, there is that. There was a difference in time, but I'm not talking about that.
Yeah, no, it's a new world. I'm not sure about it either, but it looks a very interesting association. Brilliant. Thank you ever so much, Jimena.